Imagine a detective walking into a burning building, finding a firefighter holding a hose and just arresting them for arson.
Right. It makes no
It's
sense.
completely absurd, right?
Yeah.
But in 1892, some of the world's most brilliant scientists did exactly that to a microscopic bystander.
They really did.
They blamed it for a global catastrophe. And that mistake, I mean, it cost us decades in the fight against one of the deadliest pandemics in human history.
It is a stunning historical detour. So we are pulling from a massive stack of clinical and historical microbiology sources today to trace the story of an organism called Haemophilus influenzae.
Yeah, and the mission for this deep dive is to figure out how a single tiny bacterium pulled off, well, the ultimate case of mistaken identity.
Exactly. We're looking at how it relies on a brilliant biochemical party trick just to survive, and how its microscopic anatomy dictates whether it gives you, a common earache or a fatal infection.
I have to say, looking through these sources, the sheer scale of the deception is just mind-boggling. You expect history's greatest villains to be massive, world-altering figures, not something that can hide on the head of a pin.
Yeah, well, to really grasp the impact of this bug, we have to start with its name,
Yeah.
because the name Haemophilus influenzae is, at its core, a fossilized lie.
Wow. A fossilized lie.
The influenza part is just a permanent monument to the crime it was falsely accused of.
Okay, so take us back to the scene of the crime. How does the entire scientific community get something this big so wrong?
We have to travel back to 1892 in Berlin. We are at the Institute of Robert Koch, who is essentially, one of the founding fathers of modern microbiology.
Oh, yeah.
And a German bacteriologist named Richard Pfeiffer is investigating the Russian flu pandemic. This swept the globe from 1889 to 1890.
Which was an era when steamships and trains were making the world smaller, right? So diseases were suddenly traveling faster than ever before.
Yes, exactly.
That must have been terrifying. High stakes, massive global panic, and the pressure on these scientists to find a culprit must have been immense.
Oh, the pressure was unimaginable. And Pfeiffer, he starts examining the sputum and nasal secretions of these flu patients. In almost every severe case, he keeps finding this specific organism.
Okay.
In the literature, it's described as a small, gram-negative coccobacillus that is highly pleomorphic.
Let me stop you right there for a second. Pleomorphic. Does that mean it's like a shapeshifter? Like it doesn't have one set form?
That's the easiest way to think about it, yeah.
Yeah.
Depending on the environment, it might look like a short, stubby rhodobacillus. Or it might look almost completely round, like a coccus.
Ah, hence coccobacillus.
Right. So Pfeiffer finds this shape-shifting bug consistently in his patients, and he applies Koch's postulates.
Which was the absolute gold standard sequence of steps for proving that a specific microbe causes a specific disease.
Exactly.
But wait, if Koch's postulates are the gold standard, how did they fail him here? Shouldn't that process have ruled out a false suspect?
Under normal circumstances, yes. But Pfeiffer observed such a strong, consistent correlation that he felt totally confident declaring he had found the definitive cause of influenza.
Wow.
He names it Bacillus influenzae. And because of his prestige, and, well, Koch's prestige, for decades the global medical community just accepted it. Everyone called it Pfeiffer's bacillus.
And this wasn't just some quirky academic error. This had horrifying real-world consequences when the 1918 influenza pandemic hit, didn't it?
The consequences were catastrophic. The 1918 pandemic killed an estimated 50 million people, maybe more.
It's hard to even wrap your head around that number.
It is, and as those millions were dying, military and public health laboratories across multiple continents were pouring all of their precious time and resources into frantically developing bacterial vaccines against Pfeiffer's bacillus.
So they were trying to build a wall against a bacterium while a virus was literally tearing down the house. They were fighting the completely wrong enemy. Exactly how did this happen, though? If this bug wasn't causing the flu, why was Pfeiffer finding it in the lungs of nearly everyone who was dying from it?
We now know that the influenza virus acts like a bulldozer. When a person catches the actual flu virus, it rapidly strips away the ciliated respiratory epithelium.
So the tiny hair-like structures and protective lining of your airway that sweep away debris.
Yes.
And so because the defenses are down, something else moves in.
Yeah. The sources note that Haemophilus influenzae is a strict human commensal.
Meaning?
That means it naturally hangs out in the nasopharynx, the upper throat behind the nose, of a large fraction of completely healthy people. It doesn't bother you at all. It just lives there.
Oh, I see.
But once the viral bulldozer destroys the airway's protective lining, this harmless resident exploits the opening. It invades deep into the lungs as an opportunistic secondary pathogen.
So it causes secondary bacterial pneumonia, which, if I remember the history correctly, is actually what ultimately killed a massive share of the victims in 1918.
That's right.
It wasn't the flu virus that dealt the final blow. It was this opportunistic bug.
That is the real tragedy of the situation. Pfeiffer was culturing a real, very dangerous pathogen. He just fundamentally misunderstood its causal role.
He thought it started the fire, when in reality, it was just walking through the door the virus had kicked open.
Exactly.
So how did we finally catch the real culprit? When did the scientific community realize the flu was viral?
It took roughly 4 decades. In 1933, a team of British researchers, Wilson Smith, Christopher Andrews, and Patrick Laidlaw, they made a massive breakthrough. They were working with ferrets.
Ferrets. That feels incredibly random.
It was a bit of a fortuitous accident involving laboratory animals actually getting sick from the research.
No way.
Yeah, but once they knew ferrets were susceptible, they took infected human throat washings and passed them through incredibly fine microscopic filters.
And those filters were designed to trap bacteria.
Yes. The pores in the filters were so small that any bacterium, including Pfeiffer's bacillus, would get trapped. But when they infected the ferrets with the liquid that passed through the filter, the ferrets still got the flu.
Which proved, unequivocally, that the true pathogen was a virus, an infectious agent much smaller than any bacterium.
Exactly.
Yet the name stuck. Even though the sources note there was a taxonomic update in 1917 that correctly moved it to the genus Haemophilus, the species name influenza just became permanently embedded in the Latin binomial.
Yeah, we are stuck with it. But if the influenza half of the name is a historical accident, the Haemophilus half is 100% accurate.
Oh, right.
And it reveals some incredibly strange things about how this bug actually survives.
Right, because Haemophilus translates roughly from Greek and Latin as blood-loving. And when we look at the biochemistry in our sources, this bacteria is portrayed as like the ultimate fussy eater.
Oh, fussy is an understatement. Biologically, it's a facultative anaerobe.
Meaning it can survive with or without oxygen.
Correct. Which is a huge evolutionary advantage if you are a bacteria trying to survive in deep, oxygen-depleted pockets of a human lung or a blocked ear canal.
Makes sense.
But despite that adaptability, it has a glaring biochemical weakness. It lacks the internal enzymes required to synthesize 2 vital compounds that most other bacteria can just easily make for themselves.
And what are those?
It absolutely requires an external source of hemin, which clinical labs call factor X, and NAD or NADP, which is called factor V.
Okay, so it can't make factor X or factor V. Where does it find them? In the human body.
They are locked strictly inside your red blood cells.
So if I'm a microbiologist today and I have a patient sample, and I try to grow this bug on standard sheep blood agar, which the sources say is the workhorse testing medium in labs, what happens?
Literally nothing happens. It won't grow.
Really? But there's blood right there.
Yeah, but the red blood cells in standard blood agar are fully intact. The factor V is locked away inside the cell membrane where the Haemophilus just can't reach it. To get it to grow in a lab, microbiologists have to use something called chocolate agar.
I was so disappointed when I read the sources on this.
Yeah.
Because chocolate agar contains absolutely no chocolate.
Sadly, no.
Yeah.
Chocolate agar is just standard blood agar that has been gently heated up until the red blood cells lyse, meaning they burst open.
Okay.
When they burst, they release the factor X and the factor V freely into the surrounding medium. The heating process turns the bright red blood into a dull chocolate brown color, which gives it the name.
Got it.
And once those nutrients are free, the Haemophilus thrives.
But what if you don't have chocolate agar? Because
Yeah.
earlier you mentioned labs have this like 130-year-old party trick to identify it.
Yep.
The sources call it the satellite phenomenon.
Yeah.
How does this work?
Oh, this is one of the most elegant visual tricks in microbiology. If you streak Haemophilus onto plain, unheated blood agar, as we establish, it won't grow. But if you take a completely different bacteria, specifically Staphylococcus aureus, and streak a single line of it right down the middle of that same plate, something magical happens.
What does the staph do?
The Staph aureus produces enzymes that actively break open the red blood cells for its own purposes. And as it does, it excretes NAD factor V as a byproduct.
So the Haemophilus just sits there, waiting for the staph to smash open the blood cells and then it eats the leftovers.
That is precisely how it survives. The Haemophilus will grow, but only in these tiny, delicate dewdrop colonies clustering in a tight orbit directly around the staph streak.
Wow.
It creates the satellite pattern of growth.
Hmm.
Long before modern automated biochemical testing panels existed, a doctor could just look at that little cluster of freeloading dewdrops on a plate and instantly know they were dealing with Haemophilus.
Unbelievable. Just a complete freeloader.
Pretty much.
But, okay, knowing how this thing feeds in a petri dish is one thing. If you are listening to this, you're probably wondering how it actually attacks the human body. Because our sources make it very clear that there are essentially two completely different versions of this bug, and they cause very different problems.
Yeah, everything regarding how this bacterium causes disease hinges on a single microscopic structure. The capsule.
The capsule.
Oh, right. We have to look at what's called the capsule divide. The species splits cleanly into two main branches. Encapsulated strains, which are historically typed by the letters A through F, and unencapsulated strains, which are just called non-typable H. influenza or NTII.
Okay, let's look at the worst one first. The encapsulated threat. The sources point specifically to type B, or Hib. Why is this one so dangerous?
Hib is coated in a thick, sugary outer layer. The capsule. Specifically, it's made of a polysaccharide called polyribosylribotyl phosphate, or PRP.
Okay, PRP.
You can think of this PRP capsule as an invisibility cloak or a suit of armor. It allows the bacteria to completely resist phagocytosis.
Phagocytosis being the process where your white blood cells, your immune system's frontline soldiers, basically try to grab the bacteria and eat it.
Yes. The white blood cells try to engulf it, but the capsule makes the bacteria too slippery, too well hidden.
Oh, I see.
Because it evades those white blood cells, Hib can survive and multiply in the bloodstream, and once it's in the blood, it can travel anywhere.
Which is terrifying.
It is. It tends to seed severe infections in the meninges, the protective lining of the brain and spinal cord, as well as the joints, the bones, and the epiglottis.
The statistics from before we had a vaccine are genuinely horrifying. The sources note that Hib was the leading cause of pediatric bacterial meningitis in the United States. We're talking roughly 20,000 cases a year, mostly in kids under 5.
The burden was immense, and beyond the high fatality rate, the aftermath was devastating for the survivors.
What kind of aftermath?
Anywhere from 15 to 30% of children who survived a Hib meningitis infection were left with profound, lasting neurological damage. We're talking severe deafness, ongoing seizures, motor impairments, and cognitive delays.
And then there's acute epiglottitis.
Yeah.
The way the sources describe this, I mean, if you are a parent listening to this, it is literal nightmare fuel.
Acute epiglottitis was an absolute pediatric emergency. You would have a toddler who was perfectly healthy in the morning, and by the afternoon, they develop rapid, dramatic swelling of the epiglottis, the small flap of cartilage in the throat that stops food from going into the windpipe.
So their airway is essentially just swelling shut.
Rapidly.
Wow.
The child would present with severe drooling because they couldn't swallow their own saliva, a muffled, so-called hot potato voice, and they would instinctively sit in a rigid, upright tripod posture.
Just leaning forward on their hands, trying to keep their airway mechanically open.
Exactly.
I was reading about how doctors diagnosed it, and they had mentioned the famous thumb sign on an X-ray. What exactly does that mean?
If a doctor took a lateral neck X-ray and X-ray from the side, a normal, healthy epiglottis looks like a thin little finger. But a Hib-infected epiglottis was so massively swollen that on the X-ray, it literally looked like the shadow of a human thumb pressing down on the child's airway.
Oh my God.
Before the vaccine, Hib caused the overwhelming majority of these terrifying epiglottitis cases.
Okay, so that's the encapsulated version, an absolute lethal ninja in the bloodstream. But then we have the unencapsulated version, NTAI. If we take away the capsule, does that mean the bacteria is harmless?
Not harmless, just restricted. Taking away the capsule changes its entire career path.
How so?
Without that armor, NTII gets eaten by white blood cells if it tries to enter the bloodstream. So it essentially never causes meningitis or sepsis. Instead, it gets trapped in the local mucosal surfaces.
It just wreaks havoc wherever it's currently sitting.
Exactly. It joins a triad of notorious bacteria alongside Streptococcus pneumonia and Moraxella catarrhalis that are the primary culprits behind acute otitis media.
Childhood ear infections. So if you're listening to this and you've ever dealt with a screaming toddler pulling at their ear at 2 a.m., there is a very good chance you are dealing with the unencapsulated version of this exact bug.
A very high chance, yeah. It also drives chronic sinusitis, and critically, it's a massive problem for adults with COPD, chronic obstructive pulmonary disease.
Oh, really?
Yeah, NTIT colonizes those previously damaged airways and periodically flares up, causing severe bouts of acute bronchitis.
Okay, if Hib was causing 20,000 cases of meningitis and deadly airway swelling in toddlers every year, we obviously needed a vaccine. Why was it so hard to make one? Because the sources talk about a massive failure in the 1970s.
Yeah, the first attempt to make a vaccine failed for a deeply fascinating immunological reason. In the 1970s, researchers successfully purified that PRP polysaccharide, the material that makes up the capsule, and they used just that pure sugary coating as the vaccine.
Which makes sense on paper. Show the body the armor so it knows what to look for.
It does make sense, and they tested it in a landmark clinical trial in Finland. It actually worked decently well in older children and adults. But in infants under 18 months, the exact group at the highest risk of dying from meningitis, it failed almost completely.
Okay, wait. You lost me a bit there. If the vaccine has the exact material from the bacteria's capsule, why wouldn't the infant's immune system remember it? I thought that was the whole point of a vaccine. What is it about a polysaccharide that makes the immune system develop amnesia?
It comes down to a foundational rule of how immunology works. Pure polysaccharides, like the sugars in the Hib capsule, are what we call T-cell independent antigens.
Okay, break that down for me. B cells, T cells, how do they interact here?
Think of your B cells as the factories that pump out antibodies. Think of your T cells as the generals that coordinate the long-term war strategy.
Yeah.
When an adult gets injected with the pure sugar polysaccharide, their mature B cells can react to it and pump out some antibodies, but sugars do not activate T cells.
So the generals never get called to the battlefield.
Right. And without the generals directing the response, an infant's immature immune system just mounts a weak, short-lived defense. Most importantly, a T-cell independent response creates zero immunological memory.
Wow.
The infant's immune system literally forgets the bacteria the second it's gone.
So how did they fix it? Because obviously we don't see 20,000 cases of Hib meningitis today.
It took a brilliant innovation by researchers, most notably John Robbins and Rachel Schneerson at the NIH. They created the conjugate vaccine.
Conjugate vaccine.
Right. They realized they needed to get the T cells involved, so they took that tricky PRP polysaccharide and they chemically linked it, they conjugated it to a carrier protein.
A protein from a different bacteria entirely, right? Like tetanus or diphtheria toxoid.
Yes, or a related non-toxic protein called CRM197. By physically tying the bacterial sugar to a recognizable protein, they fundamentally changed how the infant's immune system saw the vaccine.
Because proteins do activate T cells.
Exactly. Proteins do activate T cells.
Oh, I see. It's like attaching a giant neon attack me sign to a stealth bomber. Suddenly, the infant's generals can actually see the threat, they mount a massive coordinated attack, and most importantly, they write it down in the history books for next time.
That is the exact mechanism. They successfully converted a weak, forgetful T-independent response into a strong, memory-forming T-dependent response.
And what were the results?
Staggering. Once these conjugate vaccines were licensed in the U.S. around the late 1980s, invasive Hib disease plummeted by over 99% in just over a decade.
99%. That is an incredible triumph of medical engineering.
It changed pediatrics forever. And this conjugation, trick-linking a hard-to-see sugar to a visible protein, became the foundational template for entire new families of vaccines.
Really?
Oh, yeah. The pneumococcal conjugate vaccines like PCV13 and the modern meningococcal vaccines, they all rely on this exact same underlying biology. Hib was the proof of concept.
Okay, so we put a neon sign on Hib and practically wiped it out. But earlier you said the other version, NTII, doesn't have a capsule. So the conjugate vaccine doesn't work on it.
That's the lingering reality of this organism. Because NTII has no capsule, it has no PRP polysaccharide. The target for the vaccine simply isn't there.
Wow.
So while Hib disappeared from vaccinated populations, NTII remained virtually untouched.
So by knocking out the lethal ninja, we basically just cleared the stage for the unencapsulated version to become the undisputed main problem.
Yes, unfortunately. NTII is still out there driving millions of antibiotic prescriptions every year for those ear infections and sinusitis we talked about. And it remains a major factor in fatal hospitalizations for COPD patients globally.
That's rough.
Researchers are still desperately hunting for a protein-based vaccine target that could work against it, but it's incredibly tricky because NTII is highly variable.
Which brings up a question about treatment. If the unencapsulated NTII is causing all these ear infections and surviving in the lungs, why can't we just nuke it with standard penicillin like we do with other common bugs? I know people sometimes assume that because a bug is gram-negative, it's just universally resistant to everything.
Well, that's a dangerous oversimplification. H. influenza isn't a universally resistant superbug. However, many strains have acquired a very specific testable resistance mechanism. They produce an enzyme called beta-lactamase.
And what does that enzyme do?
It acts like a pair of molecular scissors. It physically cuts open the core chemical ring of ampicillin, amoxicillin, and related penicillin-style drugs, completely inactivating them.
So it literally breaks the antibiotic before the antibiotic can break the bacteria.
Yes, and because this beta-lactamase trait has become so common globally, it has forced doctors to change their frontline empiric antibiotic choices for ear and sinus infections.
What do they use instead?
We often have to use combination drugs, like amoxicillin clavulanate, where the clavulanate acts as a shield to distract the beta-lactamase enzyme while the amoxicillin goes in for the kill.
Man, it's an endless biochemical chess match.
Yeah.
And speaking of the global picture, our sources mentioned that the victory over Hib isn't entirely complete everywhere, right?
That brings up a critical point about public health. The Global Alliance for Vaccines and Immunization, Gavi, is still treating the Hib vaccine as an ongoing priority worldwide.
Because a medical problem is only solved where the intervention actually reaches the people.
Exactly. In regions lacking robust, funded pediatric vaccination schedules, Hib still causes massive suffering.
And even where we do have the vaccine, nature hates a vacuum. The sources mentioned that since we knocked out type B, some of the other encapsulated strains, types A, C, D, E, and F, are starting to make moves.
We are closely monitoring them. Type A, for instance, has drawn renewed attention because it's causing higher rates of invasive disease in some North American indigenous communities.
Wow.
It's a sobering reminder that pathogens are always adapting to the selective pressures we put on them. We changed the environment by eliminating Hib, and now the lesser-known cousins are trying to fill the void.
All right, to bring this all together, we've tracked Haemophilus influenzae from its origin as a falsely accused pandemic arsonist to its life as a biochemical freeloader waiting on staff to feed it.
Right.
We've seen how its slippery capsule made it a lethal pediatric threat, and how a brilliant immunological trick conjugating sugars to proteins all but wiped that specific threat out, leaving behind its unencapsulated chronic nuisance sibling causing earaches and bronchitis today.
It is a masterclass in how deeply understanding the precise mechanical biology of a pathogen is the only way to defeat it. But as we wrap up, I want to leave you with one final fascinating connection from the sources to mull over regarding evolution.
Oh, I always love these. What do you have for us?
We've spent this entire deep dive talking about the respiratory tract, right? Earache, sinusitis, COPD, airborne droplets, pneumonia, epiglottitis.
Right, all above the chest.
Well, there is another bacteria in this exact same genus. It's called Haemophilus ducreyi.
Okay.
It shares the exact same genus name, and it has the exact same fussy, blood-loving nutritional requirements. It relies on those exact same factors from red blood cells to survive. But it has absolutely zero clinical relationship to the respiratory system.
Wait, where does it live?
It causes a sexually transmitted genital ulcer disease called chancroid.
Wait, what?
It does. So the question I want you to ponder is this. The exact same genetic, dietary restrictions and vulnerabilities end up evolving to colonize such wildly different human real estate.
Wow. From the lungs and the ear canal to an entirely different geography. That is an incredible evolutionary leap to think about. Thank you for joining us on this deep dive into a true microscopic master of deception. Keep exploring, keep asking questions about the microscopic world around you, and we'll catch you on the next one.